All right, I'm going to try to cover more on electrical signaling, and I'm probably not going to wrap things up. Actually, I was thinking I might, but I might not. I think I can finish electrical signaling and give you a good idea, an overview of all the types that there are. That's the main goal of this one lecture. But to really tie things up, I think what I might do is just have one more where I kind of do an overview, where we really are comparing and contrasting the chemical versus electrical. Maybe. But for now, let's just start there with the review. So you remember, it's all about, a lot of this is about signal transduction, where the main thing we're trying to do here with regard to signaling is when we're talking about what does signaling actually mean, in this case, we're kind of defining it in one way. There's a lot of ways you can do it, but we defined it kind of in a way where Mother Nature created the central problem of creating a cell membrane to protect the inside of a cell or the inside of a body from the nasty environment, and to have homeostatic control over all the chemical reactions. But then it was also at the same time sort of, if you're building a house, then you need doors and windows to be able to see, what's going on outside or to get things in and out. It's very similar. That analogy is actually not so bad. And receptors do a lot of that job. And a lot of signaling then is letting the cell know, even if it's a single cell, outside of that cell is its environment, or if a cell's in a multicellular organism, outside is going to be the blood or the state of that organism's, the organism's environment. So it needs to know about that to be, a good worker in working, doing something functional for a whole organism. So signal transduction is really letting information get from outside to inside of a cell, and for cells to be able to regulate that process. And it became a problem once cell membranes were invented. For the neurons, there's a couple ways. We are focusing on a couple ways to do that. They always involve having some protein machine, some protein threaded through that plasma membrane. That's these guys. And a lot of times, because these big proteins can bind things, either drugs or natural things that the body makes to send signals, they're called receptors. And we learned that there's a couple types. And for chemical signaling, we learned that there was one type that does chemical signaling where, let's say, the receptor binds to a neurotransmitter. We were using that as an example of information that cell number 2 needs to know, that cell number 1 is trying to talk to it, so it's spitting out some neurotransmitter in this space. This is outside information that this cell needs to know about if the name of the game is communication. So it would just get around the problem by putting a receptor on its cell membrane, letting the outside part of that receptor stick to the molecule, and letting a conformational change find its way, just simply a structural change in the machine that's induced by the binding of this thing on the outside. That gets transmitted just straight up structurally, by the bending and twisting and torquing of the protein. They're flexible, they're not rigid structures, to some event that can now be read on the inside. And for chemical signaling, that readout is all done by biochemical mechanisms or chemical reactions. That was one way to do the signal. The other way to do the signal was to let the molecule, like in another type of receptor, to let the molecule pop open an ion channel where literally ions were able to flow through, like plus charges or negative charges. In this case, it looks like it's plus charges moving through, and you're getting an electrical signal, like a signal of positive charges coming into cell number 2. Now, another thing we haven't really talked about, and I guess I will have to make more lectures to give you an idea, is it just begs the question. These questions just go one after another, but just saying there's positive charges flying into cell number 2, if you're really thinking ahead, you're going to be like, so what? How does that lead to a meaningful change in cell number 2? For chemical signaling, we answered that. I gave you from start to finish an example of how biochemical reactions can lead to meaningful change in different properties in neuron 2. For electrical signaling, we're still a long way from that, but we're getting what we need. We're getting a foundation to begin to go there. But again, just saying there's a bunch of positive charges now in cell number 2 isn't good enough. That we haven't really given cell number 2 any new tricks or any up its sleeve in terms of what it can do with that information. So I think I am going to have to make more lectures. But anyway, there's two kinds of receptors. Some can do chemical reactions, And those receptors tend to be called metabotropic. I don't know if I introduced that term in any of the other lectures I gave. And these ones are called, they're doing ions, electrical signaling. Oops. Electrical signaling are doing, they're letting ions flow through, so those are called ionotropic. So there's metabotropic is doing the chemical reaction. So you would say this receptor that binds, this is a serotonin receptor, and it's a metabotropic receptor. You would know then that that's binding serotonin out here, and it's leading to chemical reactions. There's other type of receptors that are ionotropic. So if I said I had a serotonin ionotropic receptor, that would mean you've got a completely different receptor here. It's common in the sense that it can also stick and bind to serotonin, and that serotonin is leading to a conformational change. But that serotonin, that neurotransmitter, is leading to a conformational change that opens up an ion channel that's letting in, let's say, sodium ions. So ionotropics, so electrical signaling, and that's done through ion channels. Okay, so kind of bringing ourselves up to speed on this big review. That's what we got so far. But one thing I didn't answer is, like, we never really answered, like, why the flow. So remember, like, if this lipid bilayer sheet, all this stuff in here is lipid bilayer, it can't let sodium ions in. But I also told you that there's sodium ions everywhere. So if this is outside the cell, and this is inside the cell, and there's sodium channels everywhere, like I said there were. And let's say we've got an ionotropic receptor in here, so we can gate it with something. So let's say we have a neurotransmitter, let's say serotonin, and it can bind here and open up the ion channel. Ion channels got a problem because they are actually channels. The conformational change, like if serotonin binds this, right, the channel would move into some state where it's now open, where it can let sodium ions flow through. But it's straight up, it is just a pore. You wouldn't have lipid in the middle of it. So it literally is a water-filled pore where sodium can flow in. But how would you get a current? Like, there's a little problem here if you're really, really thinking ahead and you're realizing that there's sodium everywhere. You know, why isn't it the case that the sodium goes out? Like, if there's sodium everywhere, you really wouldn't have a net current flow. It wouldn't be like you'd get all the positives coming in because there's sodium everywhere. And that's sort of a central problem I haven't really talked about that I do want to talk about. And so it also involves something that's going on with the bilayers. Like, why the flow? And it's not just why the flow. Like, if you had sodium everywhere, you'd get for every sodium atom that would want to fly in from the outside, you'd be equally likely to have one from the another one from the inside, just, going the other way. And if you had a voltmeter or, an ammeter, like if you had some way to measure current, you'd get no net flow. You'd have as many moving in as moving out, and you wouldn't even you would get no signal. That's a problem. So instead of saying why the flow, it's really a question of why the directional. Why the directional flow? Like, what is the neuron doing? What magic is the neuron doing where it's letting directional flow? And if I just asked you that question, there's a couple things you might think of. Like, what would cause, let's say, the sodium to go, let's just say, and I think in the example I gave you last time, I said that the sodium currents tend to be inward. That means they go from outside to inside. So the dominant flow is this way. If I had to ask you to pause this and think about why that might be, you might come up with things that are, you will come up with things that are reasonable. And there's a real answer to one of these things. One of the things you might say is that, well, these proteins are only somehow allowing unidirectional flow. Like, the gating is a gate that somehow can distinguish inward flow from outward flow. And literally some ion channels can do that. They can let flow be in one direction versus the other. That's more of an advanced topic on ion channels. But that actually can happen to an extent, but that's not the dominant mechanism. But when that does happen, that's called rectification. And if ASA is watching this, if I'm sure you know about, like, some of your currents and circuits can exhibit this property too. A rectifier is something that allows current flow preferentially in one direction over another. So any electrical engineer knows that, like, that's just a general property of some materials and some, like, electronics, where you can actually design rectification in there where flow just goes better in one direction than another. But again, that's not the dominant biological mechanism. I'm going to tell you what's going on in the dominant biological mechanisms about why the flow. We're answering this question about why do we get, like, a very controlled directional flow, which is really, that's more of a signal than a random one going in, one going out, that's not much of a signal. So why the flow? And that involves another protein machine. And that protein machine is generally, it's a pump. It's called a pump, and there's a lot of protein pumps. And again, none of this should be surprising. Like, these, you should be learning by now that the cells can do all sorts of tricks. And if a cell is wanting to do a trick, it's going to make a protein, because the proteins are the machine. And the proteins can move and carry things, and they have all this chemistry to them where they're flexible, and they can bend and torque and exert forces, and because of that, they literally can be classical mechanical machines. And one class of these machines that can exist in plasma membranes are called pumps, and they do just that. So they literally take energy, they require energy. So I'm just going to say we need to put energy in. And that usually comes in the form of a molecule called ATP, adenosine triphosphate. But they usually, like, do a chemical reaction and get some. They, like, do a little, like, of a reaction where they can harness some of the energy coming out of that reaction to do some work. And I'm talking in these terms. I'm not using just in the English sense, I'm using in the physics sense. They can actually do electrical work or mechanical work, but they can interconvert energy types like chemical energies into literally mechanical or electrical work. Straight up physics energy interconversions. And so what they do is there's a class of pumps that tends to like to pump. They use the energy to pump out the sodium. So I was telling you that there's sodium ions inside and outside of a cell. That's true. But it's not equal. And the dominant mechanism is that there's a lot more sodium ions outside the cell. Remember, I was just drawing it so that this was the outside and this was the inside of a cell or a neuron. And here's let's say your ion channel that can gate and open. There's a lot more sodium ions outside than inside, so that when this ion channel opens, and it is just a channel, it's just like a straight up, like, little stream that these sodium ions can flow through. It's just like food dye. It's just like putting a little bit of dropping food dye in a beaker of water. The food dye always goes from high concentration to low concentration by diffusion, and they just go down concentration gradients. So that's exactly what's happening. If this ion channel gets gated by a neurotransmitter, so neurotransmitter binds on the outside, this channel is now open and it provides a path for sodium ions. Yeah, you could get diffusion out and you could get diffusion in, but the rates of diffusion are different because the concentration of sodium ions are higher on the outside because the cell is literally pumping them out and it needs energy to do that. So literally, the cell is creating like a little battery. It's creating like a little highly positively charged in terms of sodium outside environment. That's like a battery across this membrane. And when this ion channel opens, it's like turning on the flashlight. You now provide a current path. That battery has all those positive charges that you've shoved over to one end of the battery are now just going to diffuse through and create an ion current, and that's exactly what happens. So you get a sodium current, and it's dominantly inside simply because you've got a lot more flow going from out to in than you do from in to out, and that's the trick. Why the flow? It's a concentration gradient that is literally created by using the energy of ATP across cell membranes. So and almost all cells have a gradient set up a gradient of ions across their membranes and can do various versions of electrical signaling. It's really cool. So that's why the flow. All right, so in this picture now, when I was telling you, so if you put like some neurotransmitter in here so that you gate, so that now you let this ion channel in this patch of membrane that you've opened up, and now you can record it through this circuitry. You can record it and watch on a computer, like, you know, you can record this pipette to circuitry. When you put neurotransmitter in and gate this channel, you open it up, you get a dominant flow. This figure was sort of cheating. They were just showing you as if there's only sodium on the outside, but now you know it's a little bit more complicated than that. You'd have to have a lot of sodium inside this pipette and only a little bit on this side so that you'd get a net flow inside, and then you would record a net current inside. So now we're kind of getting a more sophisticated in the reality of what this like electrophysiology experiment would do when we're actually recording from ion channels. One more thing that I have not told you that I'm going to introduce now, and then we're going to be, all I want to do is introduce this main topic now on electrical signaling and kind of be done with this segment, because there's been a lot of information here too. But remember, gating is about what pops open ion channels. So what gating is about, opening the gate. Opening the gate. And I've told you that neurotransmitters can open the gate. So here I was always saying, we're putting a neurotransmitter, it binds, the ion channel pops open, you get current flow in. But now I'm telling you there's two. There's something else that can gate the channel. What is it? So in this other, I will erase. There's another thing I can do here. I can actually have no neurotransmitters at all in this pipette, and I can do a trick to open up this channel. And because this, remember, because this pipette is wired up to circuitry and electronics, I can do that. Here's what I can do. Without any neurotransmitter, if I put a voltage across this plasma membrane by using this pipette, because it's wired up, I actually have like literally like wires and electronics hooked up to this pipette, I can throw a voltage. I can actually throw a voltage, like an electric field, across this cell membrane, this plasma membrane, and without any transmitter, the ion channels can feel, they can sense and be gated by an electric field, and those are called, so we know that that you can gate two ways now. You can gate chemically with a neurotransmitter. This is brand new. This is really cool stuff. Or we can totally gate by voltage, and sometimes that's called electrical gating. And again, we can do that through the pipette. No neurotransmitter at all is needed. And if you can just throw it, all of a sudden you just put a voltage across this thing and boop, these ion channels just pop open. And it is not, like always, remember, I've always been telling you that every time like a protein is having a new trick, it usually doesn't mean it's one protein that can do many things, because that's hard for Mother Nature to build. So there's a whole separate class of ion channels. Some of them are gated chemically, some of them are gated by voltage. And this is confusing, because don't confuse a chemically gated ion channel. These are all ion channels. Don't confuse a chemically gated ion channel from a metabotropic receptor, where typically like a neurotransmitter, metabotropic, they're open, they're sort of gated by, but it's not gating because you're not like opening an ion channel gate, but they're activated by neurotransmitters. Neurotransmitters activate metabotropic receptors to do biochemical reactions. We learned about that. But also neurotransmitters can, so that's called activation. It's not called gating. So I'm going to put activation in quotes here, where they can induce biochemical cascades. But beginning students of neuroscience always get confused, because they're like, they kind of know somewhere in their brains that chemical activation, there's two kinds of them. And they always mistake like metabotropic receptors from chemically gated ion channels, because there's similarities and differences. The activation of both of these types is done through a chemical or a neurotransmitter, but the output of what that receptor is doing from that, from that conformational change is very different. This is what a metabotropic is going to activate a biochemical cascade. There ain't going to be no ion flows through a metabotropic receptor, and that's by definition. But in a chemically gated ion channel, we now call it gating, not activation. Although even still, like electrophysiologists will say activation of the gate. So there's a lot of like crossover here, but it's a completely different channel type. It's an ion channel that allows currents to flow through. You can open them two ways, chemically or through a voltage. And it's really cool. So if you look at a recording here, so this is like a recording strip of so here's the stimulus. This blue line is a stimulus. And here they were recording from a voltage gated channel. And they were doing it, by the way, they were doing it right from that, like they were doing it in just this way, where let's say they had this pipette on and then they just boop, without any neurotransmitter, they just popped a voltage. And that's what you're seeing here. So remember, because the cell made itself into a battery, its resting state, it's starting out like it's some voltage that's like, it's like a little battery. The cell actually has a voltage, like, and it actually uses energy to keep like a little voltage, and that's what you see here. And then right here, the experimenter said, I'm going to flick a switch and I'm going to put a new voltage across that plasma membrane. And as soon as he did that, he held a voltage pulse, like he held that cell at a new voltage, like where he overrode like that battery essentially of the cell. And all of a sudden these ion channels are just like, whoop, okay, there's a voltage now, like I'm getting a signal, even though it was just the experimenter in the lab, putting that signal in through his glass pipette. And these channels started popping open. They sensed it. And if you look at the time scale here, like this time scale from here to here is 5 milliseconds. So from this bar, that's 5 1,000ths of a second. That means that if these openings are happening, like these openings are happening at about 1 millisecond. They're about 1 millisecond openings. So when one ion channel, like this guy, like this blue guy, is sensing a voltage shift, it doesn't just open and stay opening. You're learning, like, there's a lot of things going on. The gating is complicated. They do like more of like a flickering thing. They're opening but then they're closing, and we haven't even gone into what's making them close. That's another complicated topic. But again, because there's like zillions of them in a plasma membrane, usually, like all of them, there's enough of a big signal when all these add up that you actually can get a pretty big signal out of ion channels. But the opening is like literally just a 1 millisecond event. These proteins are just doing things like they're literally flicking, open and closed at the, at like 1/1000th of a second. They're just like their whole event, the whole beginning and end of everything they do is done. It's actually amazing. So this is like an example, but if you did another patch recording where you didn't do any voltage, where literally instead this bar represented a puffing of neurotransmitter, you would see similar like openings, like flickering openings in response to that. It's really interesting. But ion channels kind of have similar properties, whether or not they can. Or, they can also there's quite a bit of variability too. So the whole field of ion channels is, there's a whole family of them, and they're all, they're all different and similar. And a lot of people made careers of studying just ion channels. But this is what a trace would look like, a signal from one molecule in one nerve cell. And we literally have the technology to record the event, the molecular event from one flickering opening and closing of one of these things. It's astounding. And that's kind of what brain cells are doing. So but we've been like really digging deep. We've been talking about things that almost don't even make sense anymore in the context of a neuron. So I'm going to end this lecture by bringing it back. So let's just say here's our big neuron now. We're looking at the level of a light microscope. These are all the dendrites. And remember, these dendrites are probably they could get 10,000 different inputs where all these different other neurons are sending axons in and making connections, and you have 10,000 synapses in this tree. And then the brain cell needs to decide whether or not to send a pulse of electricity down this one axon. That's what this little wire is, the axon. But now you know enough about how the synapse works. We've talked about that. But now you finally know enough to talk about the signal in the axon, because you know about voltage-gated channels. So I'm going to draw this tube. So this, I'm just drawing like this little piece of this little wire. It's all over here now. This is just a tiny piece of the tube. And we have to send an electrical pulse down that wire. And this could be 3 feet long. Like if this axon is going to run down from my brain, to my spinal cord or something, I have to send a pulse of electricity, and you now know that ion channels are doing that. But it's not like you have synapses all over the place down this wire. They are voltage-gated channels. So let's see how they work. I have a voltage-gated channel here and a voltage-gated channel here, and they're threaded through the membrane. So let's say the electrical signal is happening here, where I've initiated an electrical signal where I'm getting the positive charges in, right? And it's moving inward. So this channel is closed right now. But if this channel is open, because you have an electrical signal that's now invaded, let's say all the way in from all the signaling from these dendrites have now said, yes, we're sending a pulse of electricity down, and it's happened, so that this first part of the axon is now letting sodium ions flow in. Sodium ions are flowing in through this guy. You're getting positive charges. And that's a voltage. Like literally all these positive charges, like if normally it's like negatively charged, like down this wire, right? And these guys are all closed, because they're going to only open if they sense a voltage. Closed. Now, as these positive charges invade, this guy is now sensing the positive charge. It's sensing the electric field invading in. It's sensing a voltage. So now this guy's like, okay, I'm going to do my flicker open and more. And now this is contributing to the pluses. And now this guy's going to go like, oh, okay, I feel it. I am now open. And it's going to let the sodium currents flow through. So you can see in these openings and closing, because they're happening like they're flickering at millisecond timescales, like crazy fast. This whole like sensing and opening and letting the current flow through is happening. Like this thing marches through at crazy speed, so that when you sense a need to respond to something, when you touch a flame and you jerk your muscle away, it's a fast reaction, because this electrical signaling is running down this nervous system through wires very fast. And that fast signaling is done by these voltage-gated signaling cascades down wires, down the wires we call axons. All right, so it's really interesting. So in the dendritic tree, you're getting all these electrical events through synapses. You're getting depolarizations in the dendritic tree. You're getting a lot of biochemistry happening. And all that information somehow is either going to, or it will not be going to, lead to this neuron to decide to fire off an electrical signal to let all of its partners know, hey, I'm going to be yelling at you to let you know there's something happening. And if this neuron is involved in a motor pathway where you're learning to kick a soccer ball, let's say, all these outputs are going to be involved in all the muscles that might help you kick a soccer ball. And eventually, this neuron will literally talk to muscles. It'll literally go to muscles to move your legs. And interestingly enough, that is also done through electrical signaling. That would be a whole nother lecture. But these axons literally can talk electrically to muscles to cause them to contract to move, for example. In your brain, it's talking to other neurons to set up all sorts of circuitry and electrical circuitry and do computations. That's a whole nother level of neurobiology. But this is like the purpose of this isn't to get into any of that or to provide even any insight into that, but it is giving you the foundation to at least understand like biochemical and electrical signaling, which does form the foundation of the crazy like emergent properties and more complicated stuff that the brain is doing. And honestly, for emergent properties, we actually don't even have any insight. Even I can tell you, like as an expert in the field, there's not even much insight into how all the coordinated metabotropic receptors and ionotropic receptors and all these electrical activities are doing things like making a thought or making a memory. So we know the biochemistry and the molecules and the circuit and the currents and the mechanisms of how they all work almost perfectly well. It's crazy in molecular detail, and yet that still doesn't give us any answers to any big questions. But which wasn't the purpose of this lecture, and even if it was, I couldn't because we don't have it. But it was just to give you some understanding about what we do know and the amazing complexity of the things that lead to that stuff. Who knows? Maybe we'll talk more about that in other lectures. All right, I think I'm done. I'm going to end it with that and call it good.